Hello, my name is Kathleen Ropella. I'm from the University of Michigan, and I'm going to discuss the paper entitled A Regularized Model-Based Approach to Phase-Based Conductivity Mapping Using MRI. I'll begin by describing what we're actually measuring, and that is dielectric properties. We use the letter kappa to describe the complex permittivity or dielectric constant of a material. This describes how electromagnetic waves behave within that material. In the real part, we have permittivity, and in the imaginary part, we have conductivity. And while we do have the frequency of interest omega in this expression, it's important to note that permittivity and conductivity are also frequency dependent. On the left, I'm showing an example setup of how we might measure the dielectric properties of a sample liquid, which involves a dielectric probe and a network analyzer. However, we can't use this setup in vivo. So I'm going to talk about one of the ways we can measure dielectric properties in vivo using an MRI scanner. There are two major applications for measuring conductivity in vivo. The first is its diagnostic value. There's been quite a bit of work in recent years showing that conductivity is elevated in malignant tumors, but there are plenty of open research questions about the correlations between conductivity and other metrics we might use to describe tumors. The second application is RF safety in MRI. SAR is proportional to the conductivity, and we typically limit SAR to prevent heating within the MRI scanner. By measuring subject-specific conductivity maps, we might be able to predict subject-specific power deposition, SAR, and temperature change. One way we can measure conductivity in vivo is with MR electrical property tomography, or MREPT. The complex permittivity kappa is related to the transmit RF field, or B1+. Now, typically when we want to measure the complex valued B1+, we would need one scan for the magnitude and one scan for the phase. And an important observation by Wen in 2003 stated that the conductivity of a material primarily affects the phase of the magnetic field, and the permittivity primarily affects the magnitude. Therefore, if we're only interested in conductivity, we really only need to acquire the phase, which is going to simplify our acquisition and our calculations. This brings us to phase-based conductivity mapping, which is the topic of this work. The conductivity sigma is approximately equal to the Laplacian of phi plus, which is the transmit phase, divided by the frequency of the MRI scanner, omega, and the permeability of free space, mu naught. What this says is that conductivity is proportional to the curvature of our phase maps. And I have here a representative example of the phase in the human brain. And you can see that it's generally bowl-shaped, but there are going to be small fluctuations in the curvature, which give us a spatially varying conductivity. There are two major limitations to not only phase-based conductivity mapping, but MREPT in general. The first is that we're using a Laplacian operator, which is a high-pass filter and therefore amplifies any noise in our MR signal. This requires us to use some sort of smoothing on either our phase data, the resulting conductivity maps, or both. The second limitation is that all of these equations have been derived from the homogeneous Helmholtz wave equation. The keyword here is homogeneous, meaning we're assuming that our dielectric properties are spatially constant. This is definitely not true in the human body because we have not only spatial variation within a tissue, but we have spatial variation at boundaries between two tissues with very different conductivities. This paper describes a regularized model-based approach to phase-based conductivity mapping, which we are calling the inverse Laplacian method, or simply IL method. There are three major benefits to using this method. The first is that we're going to reduce boundary errors by using masks and a nonlinear regularization term. The second is a better trade-off between SNR and spatial resolution, again using a nonlinear regularizer. And the third is that it is a fully three-dimensional approach to phase-based conductivity mapping. The estimator for conductivity using the inverse Laplacian method is given here. And we're still estimating conductivity sigma hat based on our transmit phase, phi plus. But the important part of this estimator is our inverse Laplacian operator L, which is the approximate inverse of the Laplacian operator. So this operator is modeling the physics between our phase distribution and conductivity based on that phase-based conductivity approximation I showed you earlier. However, we're rearranging the equation so that we have phase in terms of conductivity instead of vice versa. So if we break down this estimator into two parts, the first is the data fit term, which is going to ensure that our measured data on the MRI scanner matches the modeled data. So to begin, we pick a distribution for sigma, and we multiply that by our inverse Laplacian operator L to get a calculated phase distribution. We compare that calculated phase to the measured phase phi plus, and we update our estimate of conductivity based on those differences. The second part of our estimator is the regularization term, and this is going to reduce spatial variation in our conductivity estimates while preserving edges. So the regularization parameter beta is going to control how much smoothing we do. W2 is an edge mask, which prevents smoothing across boundaries between tissues with different conductivities, and I'll describe those in more detail in a little bit. And R is the regularization function, which has edge-preserving qualities as well. To begin, we did some simulation experiments using SemCAD-X, and we simulated two nested cylinders in a birdcage coil, and the outer cylinder has a conductivity of 0.59, and the inner cylinder has a conductivity of 2.14. Then we took the phase data from the simulations and we added Gaussian noise to that. The first set of experiments we did focused on the inverse Laplacian operator L. As I mentioned before, this operator is an approximate inverse of the Laplacian operator. And we calculated this approximate inverse by inverting the DFT coefficients of the Laplacian operator. However, the DC coefficient for the Laplacian operator is zero valued and therefore cannot be inverted. So to make this invertible, we had to add a small offset delta to that coefficient. In our experiments, we varied this small offset delta from 10 to the minus 2 to 10 to the minus 7. And we observed that if the offset is too close to 0, we see quite a bit of ringing in our results. Now it appears that any of these small offsets between 10 to the minus 2 and 10 to the minus 4 would be acceptable. So in the rest of the results, we use delta equals 10 to the minus 3. Next, we look at the effects of using masks in our estimator. While we only need transmit phase data to calculate conductivity, we acquire this data using a spin echo acquisition, which means we have magnitude data with reasonable contrast that we've acquired at no extra cost. We use this magnitude data to calculate masks for this problem. Mask W1 is a region of support mask, which is calculated by simply thresholding the magnitude data. In humans, it's also wise to use an angiogram to exclude any large vessels in your anatomy. This is because we get spurious phase from flow in these large vessels. Mask W2 is an edge mask, which is calculated by applying a 3D finite differences operator to the magnitude data and then thresholding the results to exclude any boundaries between tissues from our regularization term. This is used under the assumption that any changes in conductivity are going to correlate with changes in magnitude. The black and white images on the left show example masks from our simulation phantom. The top row shows mask W2, and W2 is actually a four-dimensional mask, so it accounts for edges in the horizontal, vertical, and Z direction, which helps us account for edge directionality in our regularization term. Mask W1 is shown in the second row. This mask is important because fields exist in the background of the object. However, we don't acquire MR signal in that region, so we can't model the physics between phase and conductivity in that region. Finally, the colored images show the conductivity reconstruction results, both with and without the edge mask W2. And we can see that without the edge mask, that zero-valued boundary between the two compartments is much wider than when we do use the edge mask. Next, we look at the calculated conductivity results from our proposed inverse Laplacian method as compared to a more conventional method. We have selected a weighted Gaussian filter as our comparative method. For the sake of a fair comparison, we've selected a filter width and a regularization parameter in our inverse Laplacian method, such that they match in spatial resolution. Now, it's important to note that our inverse Laplacian method is nonlinear, so while we've chosen a reasonable input value to match the spatial resolution properties, there are definitely input values for which they do not match, and that's where we're going to see benefits in our proposed method. It's also important to note that we have not necessarily optimized the regularization parameter in our method for either the phantoms or the human brain. We also weight the Gaussian filter based on the magnitude data in an attempt to match our proposed method, and that we're not spatially filtering across boundaries between tissues or material types. To compare the noise and bias between the two methods, we ran our simulation reconstructions using 100 realizations at various input noise levels for our phase data. For the noise analysis, we subtracted the mean conductivity error at each noise level from all 100 realizations and report the standard deviation of the remaining error. For the bias analysis, we simply calculate the average conductivity error over all of the realizations at each noise level. The left side of this figure shows the results from the noise analysis. We have plotted the standard deviation of the conductivity error against the standard deviation of the noise added to our simulation results. We see that our inverse Laplacian method has consistently lower conductivity standard deviation than the Gaussian filter. This is important because if we want to achieve high-resolution conductivity maps, we need to start with high-resolution phase data, which has inherently lower SNR. On the right side of the figure, we have the bias analysis results. The top row is the results for the Gaussian filter, and the bottom row is the results for the inverse Laplacian method. The left column of these four images is the results for adding the lowest standard deviation noise level, and the right column is the results for the highest standard deviation of the noise level. We observe that in both the low and high noise situations, our inverse Laplacian method does have some structured error. However, at the low noise level, we see a much narrower boundary error between the two compartments as compared to the Gaussian filter. Next, we look at the conductivity results for this numerical phantom. On the top left, we have the true conductivity profile, and in the middle, the results from the Gaussian filter, and on the right, the inverse Laplacian method. We can see that we have lower variation of conductivity values within each compartment using the inverse Laplacian method. And based on the plot on the bottom, we can see that the inverse Laplacian does have a smaller zero-valued ring between the two compartments. Next, we ran these same experiments on a Saline Phantom. These were nested plastic cylinders where the inner container was filled with deionized water, which is not conductive but will still give us a MR signal. And the outer container is filled with a saline solution with an approximate conductivity of 1.38 Siemens per meter. This is a bit different from our simulated phantom in that there is a physical plastic boundary between the two materials, which winds up getting masked out in our region of support mask, and we'll see that in the results. So similar to the last set of results, we have our true conductivity map, the results from the Gaussian filter, and from our inverse Laplacian method. And we can see that the conductivity in the outer compartment is much closer to the true value using the inverse Laplacian method than the Gaussian filter. We also see some error in the deionized water compartment, and we believe this is because there is a high noise level in that compartment and a low phase curvature due to the compartment being non-conductive. Finally, we calculated the conductivity for 4 healthy volunteer subjects. For each subject, we acquired an angiogram to use in the support mass calculation, spin echo data to get the transmit phase for our conductivity calculations, and a T1 weighted image so that we could do some tissue segmentation for further analysis. Here we have the results from a representative human subject. On the top row, we have the magnitude images from our spin echo acquisition. In the second row, we have the tissue segmentation, which we performed using SPM. It's important to note that some pixels are not assigned a tissue type, and that is because we only assigned a tissue type if the probability of the pixel being that tissue was greater than 95%. In the third row, we have the conductivity results from the Gaussian filter, and in the fourth row, the results from using the proposed inverse Laplacian method. We can see that the conductivity in the cerebrospinal fluid, which is particularly apparent in the ventricles, is much higher than the gray matter and the white matter. We also see these zero-valued black regions surrounding the ventricles, which we believe are due to boundary errors. We can also see that using the inverse Laplacian method has reduced the variation within a tissue type, which is particularly apparent within the large white matter regions. If we look at the conductivity values for all subjects across the three different tissue types, we see that the standard deviation within a given tissue type is lower for the proposed inverse Laplacian method than for the Gaussian filter. We also see that the mean values for both methods are quite a bit different from the nominal value. And that might be because of intersubject variability, but also because the apparent resolution in our segmentation images is quite a bit better than the apparent resolution in our conductivity images. So we might be blending tissue types inadvertently. Some future work for this method would include adding a non-negativity constraint to the estimator, adapting the method so that we could calculate permittivity or even the complex dielectric constant instead of just conductivity. And finally, using our inverse Laplacian model to account for spatially varying conductivities. Finally, we'd like to acknowledge our colleagues at the University of Michigan fMRI Lab, support from SEMCAD for Science for our SEMCAD license, and also funding through a GAN fellowship. Thank you.